Welcome back. Next up is Dr. Jenna Dockweiler, who's going to be presenting Canine Genetics 101 and puppy DNA testing. Dr. Dockweiler is a graduate from Kansas State University's College of Veterinary Medicine with honors in 2014 and completed her small animal rotating internship at Wheat Ridge Animal Hospital in 2015. She then completed her comparative theriogenology residency at Cornell University in 2017 and became a diplomate of the American College of Theriogenologists that year. She practiced small animal theriogenology and general practice for 4 years before becoming a veterinary geneticist with Embark Veterinarian. Take it away, Jenna.
All right. Thank you so much, Lisa, for that lovely introduction. As Lisa said, my name is Jenna Dockweiler. I am one of the professional services veterinarians here at Embark. So my main roles here are education, which of course is why I'm speaking with you all today, and also results support. So that means that if you get a result you're not sure about, or maybe it's a little more nuanced than you're used to, I can chat with you about what that means for your breeding program. So today I'm really excited to talk about some basics and also about puppy testing. So let's go ahead and get started. So here is the outline of our topics for today. So we're going to start with defining some terminology and concepts, which are just going to be important for you to understand before we dive into the bigger genetics topics. So that would include genetic diversity and genetic health testing. We're also going to chat about how to apply those results to your breeding program and why you may want to consider DNA testing your puppies as well as your breeding pairs. All right, let's talk about some genetics concepts and define some terminology first. All right, so before we really get started here, what exactly is DNA? DNA stands for deoxyribonucleic acid, which is just a big fancy word for the genetic code that's going to be unique to every individual. So DNA is hereditary, meaning that it is inherited from an individual's parents. It's organized into genes that have specific functions and lives within the chromosomes of our cells. So those genes are going to control a lot of what makes an individual an individual. So physical traits, diseases, and also some behaviors are genetic. And then I do like to mention that some people do get a little bit confused about the terms DNA testing versus genetic testing. And really, those two are the same thing. So things that we can do with the DNA or genetic test would include identification, parentage, coat colors, other physical traits, inbreeding, and also disease-associated mutations. So we're going to talk more about some of those. But I do just like to throw it out there because I know it can cause some confusion. There are some folks who have DNA testing as only identification and parentage in their heads. Next concept is genotype. So the genotype of a dog is its complete set of genetic material. That same word can also be used to refer to the allele an individual carries at a particular genetic location. All an allele is just a form of a gene. So that could be wild type, which is the naturally occurring form of that gene in nature, or a variant or a mutated form. And kind of as a general aside, wild type doesn't necessarily mean good, and mutant type doesn't necessarily mean bad. Most mammals are going to be diploid, meaning they have two sets of each chromosome. This also means they have two copies of each allele, and they're going to inherit one of those alleles from each of their parents. So because they inherit one allele from mom and one allele from dad, that means they can be homozygous, meaning they have two identical alleles of a particular gene. So that could be the wild or the mutant type. Or heterozygous, meaning they have two different alleles of a particular gene. And then lastly, I just have the word locus on here. That just refers to the physical location of a gene on a chromosome. I will use that word a few times throughout this presentation. I just wanted you to be aware of its meaning. Next concept then is phenotype. So that just refers to the set of observable characteristics of an individual that result from the interaction of the genotype with the environment. So that could refer to physical traits like coat color, disease states, behavior, even things like lab test results would all fall under this phenotype definition. So what's a gene variant then? This is a permanent change in the DNA sequence that makes up that gene. So you very, very likely remember learning this as a gene mutation. And those two terms, variant and mutation, are still used fairly interchangeably today. But we do try to use the term variant because not all of those changes are going to be deleterious or harmful. So, for example, a brown instead of a black coat color is not at all harmful to a dog, even though that is technically a genetic variant. All right, next concept is mode of inheritance. And to put this simply, this is just how a variant is going to be passed down to the next generation. So again, each animal inherits one allele from their mother and one allele from their father. So that means if we're talking about disease or trait associated alleles, a dog can have 0, 1, or 2 copies. Those are kind of our options. So you can also think of this as the number of copies of a variant that are needed to express a disease or a trait. So we've got 4 main modes of inheritance that we see in dogs. Those are going to be dominant, recessive, codominant, and X-linked recessive. So for our dominant conditions, only 1 copy of a variant is needed. So an animal only needs to inherit 1 copy from 1 parent. So of course, that also means that at least 1 parent was they themselves at risk for that disease or possessed that trait. For our recessive conditions, we're going to need 2 copies. So an animal needs to inherit 1 copy from both parents. Those parents may or may not be at risk for the disease or express the trait. It just depends on what their genotypes are. For our codominant or additive conditions, technically those are very slightly different, but for our purposes, we'll consider them to be essentially the same. Animals with no copies of a variant are not at risk for a disease or do not possess a trait. Animals with 1 copy are somewhat at risk or display some kind of an intermediate phenotype. And animals with 2 copies are most at risk or display a more quote unquote severe phenotype for whatever condition is associated with that variant. And then for our X-linked recessive conditions, male animals need only 1 copy to express the associated disease because they only have 1 X chromosome. Those male animals would also be expected to pass on that 1 copy to all of their female offspring. Female animals, on the other hand, generally are going to need 2 copies to express the associated disease because they do have 2 X chromosomes. That is not a perfect science in nature due to a phenomenon called X inactivation. That's a little bit beyond the scope of the talk, but please feel free to email me if you're interested in chatting more. And then I do want to point out that a complex mode of inheritance is also possible. You may see that on your results. So that could mean that multiple genes control the trait or genetic and environmental factors control it. Or it might be that the mode of inheritance actually is not clear based on the published evidence. So we'll go ahead and talk about that more in just a little bit here. All right, what then is genetic screening? So this is the process of testing a population for genetic diseases in order to identify individuals that either have the disease risk or the potential to pass it on to their offspring. So for us as breeders, generally we're going to be most interested in the potential to pass disease alleles on to offspring, kind of half of that definition. But we can use genetic screening in a personalized medicine sense as well. So important to know that genetic testing is prognostic rather than diagnostic. So it is only able to tell you if a dog is at risk for a disease. So even if a genetic test does show disease risk, it is not a guarantee that they will go on to develop that disease. Also really important to keep in mind that a negative result on a genetic screen for a specific disease is not an absolute guarantee it will never develop. There's always potential for a new or unknown genetic variant that's going to lead to a similar disease cropping up in a dog. So progressive retinal atrophy, which is a disease that leads to vision loss over time, is actually a really good example of this phenomenon. There are a ton of different types of progressive retinal atrophy, and not all of them are going to have genetic tests available because we haven't found what genetic variants lead to the condition in several cases. All right, and I do want to touch on this idea of congenital versus genetic because the distinction is subtle, but it's important to understand. So a congenital anomaly is defined as an abnormality that is present at birth. So those could be caused by genetic variants, could be from other causes, such as nutritional deficiencies or excesses, toxins, infections during pregnancy, that sort of thing. or they could be idiopathic in nature, meaning that they have no identifiable cause whatsoever. So a genetic disorder, on the other hand, is defined as a disease occurring in whole or in part as a result of a genetic variant or variants. So important to understand that our congenital anomalies may or may not be genetic, and our genetic disorders may or may not be congenital. So, for some examples, a cleft palate is always a congenital anomaly because that is something that is present at birth. But a cleft palate that's caused by excess vitamin A intake in the dam during pregnancy is going to be an example of a cleft palate that's congenital but not genetic. But a cleft palate caused by a variant in the DLX6 gene in a toddler puppy is going to be both congenital and genetic. And then genetic disorders may be congenital as well, although symptoms often aren't going to occur until much later in life. So, to use our progressive retinal atrophy example again, many forms of PRA aren't going to lead to vision loss until several years of age. So, that would be an example of a genetic disorder that is not congenital. So I touched on this a little already, but genetic testing is capable of showing us breed ancestry, health conditions, many of our coat and physical traits, an inbreeding assessment or coefficient of inbreeding, and we're going to talk about that in just a minute here, parentage and individual identification, and personalized medicine information. So these are genetic variants that are important for veterinarians to know, but don't necessarily affect the health of the dog. So probably the example that most people are familiar with is a condition called MDR1. And that actually stands for multi-drug resistance, but this condition actually makes dogs more sensitive to certain medications. So those medications can include chemotherapies, certain anesthetics, and some deworming medications. So dogs are otherwise perfectly fine and normal. You'd never know that they had MDR1, and many are not going to be diagnosed until they encounter one of those problem medications. But we can avoid having a reaction altogether by genetic testing and avoiding or dose reducing any of those problem meds. So I do want to touch a little bit on about how breed is determined on a genetic test. So how we do this is we're looking at very long stretches of the genome in dogs of known breeds from our reference panel. So when we see identical long stretches of DNA in two different individuals, that means that they were inherited from the same somewhat recent ancestor. So if we go ahead and add up all the stretches of DNA that match perfectly to one and only one breed, we're then able to identify how much of their DNA must have been inherited from that breed. So we then go ahead and do this for any other breed that's found in the dog. And then from all that, we can kind of understand what percentage of the dog's ancestry came from each breed. And the reason I mention it is because there are a few idiosyncrasies with this process that I do want to point out. So first, I do want to point out that a DNA test cannot determine purity, and that's largely because purebred status in itself is not a scientific designation. So that's going to include our human-defined registration status and pedigree records that indicate all of a dog's ancestors were of the same breed. Does not require, of course, any kind of examination of their genetic material. So individual dogs that are registered purebreds can and do display ancestry from a few different breeds in some cases. And these results don't affect registration status in any way. So a few reasons this might occur could be because the dog has an ancestor that is a member of a closely related breed, potentially was utilized prior to the closing of that breed's stud book many generations ago. Also could be that that dog comes from a bloodline that's geographically quite distinct from the group of our reference panel dogs. So in that circumstance, those dogs are often going to contain genetic signatures that are not common in that breed, so they can actually be useful for maintaining genetic diversity in the breed, which we'll talk about more in a minute here. So designer breeds also can raise some questions because once we get past that F1 or that initial cross of those two purebreds, the results may not be 50-50 ancestry from the parent breeds. So if we take a golden doodle example, if you breed a golden doodle to a golden doodle, that offspring may not be 50% golden and 50% poodle. And that's because each offspring does inherit 50% of their DNA from each parent, but it may not be the same 50%. So in other words, may not be 25% golden and 25% poodle. So it's also similar to note that each sibling might vary slightly from one another in their breed percentages. All right, let's next talk about some genetic variant testing. So there are going to be two basic types that we can do. Those are direct variant testing and linkage-based testing. So our direct variant tests assay the exact disease or trait-associated gene and determine the genotype at that locus. Whereas linkage tests don't test for a variant directly. This type of test is going to detect a gene or genes that are physically very close to whatever variant we're interested in on the chromosome. So those genes are expected to remain in close proximity during meiosis, which is how egg and sperm cells are produced. So they're expected to be inherited together. So in other words, the presence of that linked gene infers the presence of whatever variant that we're actually interested in. The linkage tests might have slightly lower accuracy than direct variant testing, but predictive accuracy for this type of test is typically still quite high. Polygenic conditions refer to diseases or traits that are going to be controlled by two or more genetic variants. Therefore, of course, they're not inherited in a simple fashion. And then our complex or multifactorial conditions are diseases or traits that are controlled by inherited as well as environmental factors. So a really good example of such a disease is canine hip dysplasia, which does involve really complex interactions of a dog's genetics with its environment. So both our polygenic and our complex or multifactorial diseases and traits are not going to be easily genetically tested at this time. But the good news is polygenic and complex conditions can at least be partially assessed by a polygenic risk score in some cases. So this is a score that's determined by utilizing several different genetic variants that are known to have a collective influence to assess the risk of a certain condition. So polygenic risk scores really do remain in their infancy in the dog, but they're likely to become more common moving forward. So the accuracy of direct and linkage-based disease allele testing is going to be quite high for most of our reputable genetics companies and university laboratories. But I do want to point out that there's no required regulatory oversight for veterinary genetic testing in North America. So selecting a reputable company is really, really imperative. So Embark's lab is CLIA approved and follows ISO standards. So we do meet the high quality control standards of human laboratories. All right, next we want to talk about panel versus individual tests. So a genetic testing panel is able to screen dogs for many different genetic mutations all at once. So one cheek swab is going to offer information on hundreds of different diseases and traits that are known to affect a really wide variety of different breeds. Whereas breed-specific testing generally returns results only for diseases or traits that are known to affect a certain breed. So because certain diseases may have only been seen in one particular breed, might seem silly to run a full panel of tests, but in some cases, a panel test can save you some money if there are many known genetic conditions in your breed, and particularly if you're looking for traits as well, so things like coat color. And in other cases, genetic diseases that are thought to only affect one breed might pop up in other breeds. So that means that panel testing really can serve as a canary in the coal mine, if you will, and identify diseases that are not thought to occur in specific breeds, hopefully before we start seeing affected dogs. So as with any testing, there are going to be a few specific limitations with genetic testing. First, and perhaps most obviously, if we don't know what genetic variant causes the disease, we're not going to be able to test for it. Again, diseases caused by multiple mutations are not going to be easily tested at this time. And diseases that are caused by interactions of the genotype with the environment are also not going to be easily tested at this time. Autoimmune diseases. So this is where the body's own immune system, normally that immune system functions to fight off foreign invaders such as viruses or bacteria, goes a little haywire, turns on itself, and attacks its own tissues. Those tend to pose a few specific problems. First of which, the area of the genome that controls that immune system is still not fully understood, which makes it very hard to query. And second, many dogs that do have an autoimmune disease need some kind of trigger to actually set off those symptoms. So that could be a medication, a vaccine, something else in the environment, or sometimes we never figure out exactly what it was. So it's just not possible for us to know which dogs are genetically susceptible to autoimmune diseases, but live their entire lives without ever encountering such a trigger. So that just makes test validation for this type of disorder really, really tough. Genetic testing can't tell you if an individual will develop disease nor what the disease will look like, although we do have some statistical measures that will help us interpret some of that risk and make some predictions. And with the exception of very few specific immune deficiencies, we generally can't tell which dogs are going to be more susceptible to infections or parasites. All right, let's pivot a little bit and discuss genetic diversity next. It's a pretty hot topic now that our understanding of its importance is growing. So let's go ahead and discuss what it is and how we might maintain it. So before we really get into genetic diversity, let's chat about what an ideal breeding candidate looks like. So in my mind, an ideal breeding candidate is generally healthy, has a sound temperament, passes all the breed required health testing, meets that breed standard, contributes positively to the breed, meets the goals of the breeder, and helps maintain the genetic diversity of the breed. So those are a lot of factors to consider. And so with a lot of our breeds, especially those with a small gene pool, some of these factors may not be perfectly met. So it's just important to remember that there is no perfect dog. So for me personally, the two factors that I feel really can't be sacrificed here are going to be health and temperament, because these, above all else, are going to be the characteristics that keep dogs in their homes for life. So the coefficient of inbreeding, or COI, is going to be defined as the likelihood of inheriting two identical genes from each parent. That's usually going to be expressed as a percentage, and it just represents the level of inbreeding in an individual. So the COI of an individual increases with increased relatedness of its parents. So I do like to remind folks that some degree of inbreeding is necessary to maintain those desirable, consistent characteristics and traits of a purebred breed. So it's not 100% all bad. And there are a few different ways that we can estimate it. So we can use a pedigree analysis, or we can use genomic measurements. So our pedigree-based COIs depend, of course, very heavily on record-keeping accuracy. Usually those are going to go back 3, 5, maybe 10 generations. And these pedigree-based COIs can deviate really substantially from genomic-based COIs because they assume 50% of DNA in any given offspring comes from each parent, 25% from each grandparent, so on and so forth, all the way back. That's actually not going to be entirely true in biology. That's largely due to a phenomenon called recombination. Also, founding members of a breed were often related to one another. So a pedigree-based COI is just going to never consider that, no matter how far back you go. And even full siblings can have different genomic-based COIs because, again, each sibling does inherit 50% of their DNA from each parent, but it's not necessarily going to be the same 50% from each parent. So then why do we care if I've just told you that we do need some inbreeding to maintain our breed characteristics? And we care because of a phenomenon called inbreeding depression. So this is the term for reduced fitness in the offspring of related individuals. So in the dog, reduced fertility, smaller litter size, shortened lifespan, reduced puppy survival, and increased disease prevalence have all been associated with an increased measure of inbreeding. So then what number do we shoot for? So I know this is very frustrating to hear, but the average COI varies really greatly depending on breed, and there's really no one size fits all approach to maximize the genetic diversity that we have within a population. So in other words, there's no one number I can tell you to breed towards to minimize the effects of inbreeding depression. But we do have some general strategies to maintain the genetic diversity that we have within a population. So first, it's important to know what COI you're actually dealing with in your breed. And for that purpose, I would recommend using a genomic measure because, as I mentioned before, it's going to be more accurate. But whether you're using a genomic or a pedigree-based COI, you should be able to use that information to estimate the COI of offspring of a particular pairing. And that's because the COI of the offspring depends on how related the parents are to one another, not how inbred they each are. So even two parents with high COIs might produce puppies with lower COIs if the two of them are not closely related. And two parents with low COIs might produce offspring with high COIs if they are closely related. So some breed groups do put low COI individuals into breeding homes, and that is great, but it's not the whole story. So we do need to do test matings as well. Second strategy is it's important to maintain carriers of recessive diseases within the population and slowly reduce the allele frequency over time. So these individuals aren't going to show signs of the associated genetic disease, and they can be bred smartly to avoid producing at-risk puppies. So we're going to chat more about that in detail in just a minute here. And in some cases, we can even use affected animals with recessive conditions. They can be responsibly bred, of course, provided breeding doesn't exacerbate their condition. So we're going to chat more about that too. Then, of course, another common pitfall to avoid is going to be popular sire syndrome. So this is where one sire is very overly used within a population. Perhaps he does a lot of show ring winning. Perhaps he's very available, very fertile. So doing that repeatedly is, of course, going to decrease diversity. So really, especially in our rarer breeds, we should consider utilizing all of our healthy, stable temperament individuals to help maintain that diversity. All right, let's talk about how we responsibly apply our genetic health testing results next. So in order to effectively apply these genetic testing results to our breeding program, we do have to take into account several things. We need to know the breed of dog, what variant we're talking about, how that variant is inherited, the disease phenotype or how it affects the dog, and the allele frequency of the variant within that breed or population. So the allele frequency refers to the proportion of a variant allele at a specific locus. So that's usually going to be represented as a percentage or a decimal. So it's going to range from 0.00 to 1.00. So 0.00 meaning no dogs within the population possess any copies of the variant, and 1 meaning all individuals within the population possess 2 copies of the variant allele. So this just gives us an idea of how common the disease-associated allele is within that population, and that information is really important to consider when we're taking into account genetic diversity. All right, let's talk about some of these needs from the previous slide. So why do we care about what breed we're dealing with? And that's because certain genetic variants do increase the risk of disease in certain breeds or populations, but not in others. So to go back to our progressive retinal atrophy example, there is a type of cone rod dystrophy. It's abbreviated PRA-CRD4 CORD1 in the miniature long-haired dachshund. But we've actually found that variant in Labrador retrievers and French bulldogs, but those breeds appear to be clinically unaffected. So it's not something necessarily that we would take into account for those breeds. So why does the allele frequency matter? I touched on this already, but some of these genetic variants are going to be seen at really, really high rates in certain breeds. So eliminating them from that population could result in a really, really precipitous loss of genetic diversity, which is not ideal. So I think the example that's probably top of mind for most people is an FGF4 retrogene on chromosome 12 that's associated with chondrodystrophy and type 1 intervertebral disc disease in a really, really wide variety of breeds. So in the French bulldog, where intervertebral disc disease is a clinical concern, the allele frequency of this variant is really, really high, 0.94. So that means that there are really only a few individuals with 1 or no copies of this variant. So elimination, if it is even possible to do, would have to be done really, really slowly to avoid a genetic diversity bottleneck. So that practice is going to necessitate breeding individuals with 1 or 2 copies with really, really careful mate selection. And then, of course, there are other instances where disease-associated variants are fixed within a population, so an allele frequency of 1. So eliminating that variant is not going to be possible without some kind of an outcrossing program to other breeds. And then lastly, we do want to consider the disease itself because its impact on the health and the welfare of the dog also should be considered, as well as the availability of a prevention or a treatment strategy. And that's really going to be especially important when we're considering using affected dogs in our breeding program, which again could be a viable option, which we're going to talk about now. So these are kind of generalities, but in general, for our autosomal recessive variants where the allele frequency is low, we should avoid breeding 2 carriers to one another. So doing so is going to result in about 25% of that litter inheriting 2 copies of the variant allele. So that means that they will be at risk for the associated condition. But carriers of autosomal recessive variants that have other desirable traits can and should be bred to clear or homozygous wild type individuals, because that mating is going to help us preserve that genetic diversity and doesn't result in disease in the offspring. So even an individual that's at risk for an autosomal recessive condition can also be bred to a clear individual, of course, provided they're otherwise an exceptional breeding candidate and breeding doesn't exacerbate whatever condition they have. So all of the pups that result from that pairing would be carriers of the condition, but that strategy really can preserve desired traits. The affected animal ideally should be replaced by a normal descendant when that does become possible. And then for our high allele frequency autosomal recessive variants, we're either left with an outcross program if it's needed and the variant is fixed, or breeding individuals with 1 or 2 copies of the allele with clear individuals when we can do so. All right, so here are some examples of Punnett squares. So these are charts that allow you to determine the expected percentages of different genotypes in the offspring of 2 parents. So we have a parent on the top and to the left of each chart. And because each parent passes 1 copy of an allele onto each of their offspring, we can determine what we would expect to see with each cross. So I've got black as clear, blue are carriers, and red are at risk. So our top left example here is going to be a clear-to-clear mating. Obviously, no puppies have any copies of that recessive variant because the parents also have no copies. Top right example is going to be a clear to a carrier mating. So we can see that about half the litter would be carriers, but none are going to be at risk. The bottom left example is a clear to affected. So we can see in that circumstance, the entire litter are carriers, but still none are going to be at risk. So that's why it could be appropriate to use affected individuals in your program. And then the bottom right example is going to be a carrier to a carrier mating. So we can see that about 25% or 1 in 4 puppies would be affected and 50% or 2 in 4 would be carriers. So because, of course, that mating can produce affected puppies, we want to avoid it when it's possible. All right, for our autosomal dominant and co-dominant variants in populations where elimination of individuals from the gene pool is not expected to rapidly decrease genetic diversity, individuals with 1 or 2 copies ideally should not be bred. So instead, we do like to use a first order relative that doesn't have whatever that disease-associated allele is, if one is available. But if breeding these individuals with 1 or 2 copies is necessary to maintain genetic diversity, we're going to want to give preference to individuals with only 1 copy, so heterozygotes, and they should be replaced in the program by a clear offspring when that's possible. All right, more Punnett squares. So our top left example here is a clear-to-clear mating. So again, no puppies are going to have any copies of the dominant variant because the parents have no copies. Our top right example is a clear to a heterozygote, so 1 copy mating. So we can see that about half the litter are also heterozygotes. So they are also at risk because this is a dominant condition. Bottom left example is our heterozygote to heterozygote. So 75% of that litter is at risk and only 25% are not at risk. And the bottom right is an example of a homozygote to homozygote mating. So both parents have 2 copies. So of course, all of those puppies are also going to be homozygous or have 2 copies and be at risk. So this would be what a Punnett square for our fixed variants would look like. All right, for our X-linked variants, normal males can be bred. They don't have any copies, therefore they can't pass on the variant that's associated with these diseases, so they are totally fine to use. But known carrier females shouldn't be bred because half their male offspring is going to be at risk. So usually these are variants that don't occur at high allele frequencies within breed populations. They also tend to be quite bad. So most of the hemophilias, which is a really bad excessive bleeding disorder, are going to be inherited in this X-linked recessive manner. All right, last Punnett squares in this talk, don't worry. So this one's going to be a little bit more complicated because females, again, have 2 X chromosomes and males have 1 X and 1 Y. And because these variants are carried on that X chromosome, there's not going to be a letter associated with the Y chromosome. So in these examples, the dam is listed on the top and the sire is listed on the left side of each square. So that left example there is a clear-to-clear mating, so no puppies are going to have any copies of the recessive variant because the parents have no copies. And then the right example is going to be a clear male to a carrier female mating. So we can see that about half the females in the litter are also going to be carriers and likely would not be affected, but half the males are going to be at risk because they only have that 1 X chromosome. So this is why we don't recommend this type of cross. All right, let's switch gears a little bit and chat about why you may want to consider DNA testing your puppies as well as your breeding dogs. So, there are going to be several practical reasons you might want to test your pups. So, first, if you've used multiple sires on one female, DNA testing can determine which puppies belong to which sire. If the pairing that you've done might lead to carriers of disease, or if you're looking to keep puppies back for your program that can produce certain colors or coat traits, DNA testing can also tell you which pups might be best to hang on to. And as we mentioned when we discussed inbreeding, each puppy in a litter is going to have a very slightly different genetic COI. So keeping the pup with the lowest COI can help preserve diversity within your line. As long as they're still paired smartly, we still need those predicted COIs of potential pups like we talked about. So there are also business reasons to test puppies. So puppy testing can help prove to your potential puppy buyers that you're a responsible breeder. And as DNA testing becomes more common, testing the puppies as well as our breeding dogs is becoming the new standard of care. So it does help increase your credibility and helps show owners that you care. So our surveys have shown that 9 out of 10 puppy buyers do consider a breeder who DNA tests their litters more responsible. And buyers are beginning to request this testing. Our data shows that 98% of puppy buyers surveyed would prefer to purchase a puppy that has already been tested. It can also help add value to your breeding program. So, DNA testing, of course, can be performed at any age. The DNA that's inherited from one's parents doesn't change throughout life. So, this is really the first health test that we're able to do. So, it helps you produce healthy puppies for your families. And we've also found that 96% of buyers are willing to pay a premium for those tested pups. All right, quickly before we wrap up, I just wanted to share a few final thoughts with you all. So I know that this can be really, really, really overwhelming. So if you've never DNA tested before, I'd encourage you to start with your sires, or at least the most frequently used sires. That can help you practice applying some of these principles to your program. And of course, sires typically produce more puppies than dams just because they're physiologically able to do so. If you've done a few DNA tests, I would suggest testing your breeding pairs next, or at least the most frequently used pairs. So that can help you practice health, traits, and also help you start incorporating COI data into your program. And then if you're already testing those breeding pairs, that is amazing, but you can now consider testing puppies as well. So that's both going to elevate your status according to the public and can help you make selections on which pups you're going to want to keep back for your program. So I hope you've all come away with this conclusion, but there is a ton of information that we can get from just a simple cheek swab, which really can be performed at any age. So I know this is a lot of information. It's all very complicated. If you haven't run this type of testing before, just start small and don't get overwhelmed. And Embark really is here to support you on your journey to learn to incorporate genetics into your program. And then lastly, I just like to remind everyone when I give these talks that our responsibility is to do the best we can with the available information and resources. At the end of the day, this is biology. It's not always going to be perfect. Bad things can still happen. That doesn't make you a bad person or a bad breeder, as long as we're continuing to strive to learn and improve. And with that, this is our breeder-specific email address if you would prefer to reach out privately, but I will go ahead and invite Lisa back, and I'd be happy to take any questions at this time.
Thanks, Jenna. We have a bunch of questions in here. I love your presentation. It's a great tutorial for those new to genetic testing, but it's also a wonderful refresher. I learn something new every time I watch it. So thank you. All right, let's start here. Here's a good question. Is a coefficient of 40% acceptable with rare breeds? We are working to reduce the coefficient as the population increases.
So unless you are using an outcrossing program, if you have a closed stud book and that 40% is representative of what you see in that breed, it's going to be really, really hard to actually lower that number. So whether or not it's an acceptable number really depends, again, heavily on the breed and what you're seeing. If you're seeing shortened lifespan, if you're seeing increased disease prevalence. So it's hard to answer exactly without knowing the breed. But that sounds like something that potentially could benefit from a directed outcrossing program.
Okay, here's a question. Can we repeat what we should look for in an ideal candidate
Oh,
for
yes. Would you like
breeding?
to go back to that slide? Here, let me.
Yeah, go back to that slide for a second.
All right. Hopefully everybody can see that slide. So, our ideal breeding candidates are going to be generally healthy. And when I say that, I mean not having a bunch of illnesses, as they move through their life. So not necessarily things that we can health test for, but just a generally healthy dog, not, sickly all the time. Having a sound temperament. And when I say that, I mean sound for the breed and really easy for people to live with. Obviously, a terrier is going to be very different from a retriever temperament, but something that your average pet owner can live with. Health testing would include both genetic testing and OFA testing. So ideally, we're going to pass all of those. Meeting the breed standards. So for me and my breed, that means the American Kennel Club. That might be a different kennel club for you. Contributes to its breed in some positive way and meets your goals as a breeder, and also maintains diversity of the breed as a whole. So again, there's not going to be a perfect dog. So we kind of have to balance what each of these factors are to make our breeding selections.
Okay, next question. When you have one variant of CDDY IVDD type 1 IVDD, what are the chances that our male will actually have that issue? If you choose a female that has one variant, what are the chances of the litter having a chance of that disease?
So the chance of disease actually developing varies in a very, very breed specific manner. So without knowing the breed, it's impossible to say what the chances are. The breed, sorry, my Welsh Springers are making an appearance here. The breed that has the most data behind it is actually the Dachshund. So that's those guys often have one or two copies of this variant, and about 20% of them do go on to experience back problems throughout their life. So that's the best kind of data that we have. And as far as puppies, if you're breeding one copy to one copy, about 25% would have two copies or be homozygous for that variant. About 50% would have one copy, and 25% would have zero copies. Because that is a dominant variant, the risk of having back problems actually doesn't matter if you have one or two. The risk is going to be equal. So about 75% of that litter would be at risk for potentially developing back problems. And that's a very complicated variant. There are a lot of other factors, both genetic and environmental, some of which we know, some of which we don't know, that do go into the risk of that developing.
Here's a sort of a follow-up question to that. She says, so all of my dogs carry one variant of IVDD and they are all poodles. Does that mean I shouldn't breed them? She is new to breeding.
That is also, I'm going to guess these are miniature or toy poodles. That is also very, very common in the small poodle breeds. So it doesn't necessarily mean that you shouldn't breed them, because it is so common. If we just, removed every poodle with one or two copies of that CDDY variant, we wouldn't have any poodles left to breed. So not necessarily. So I actually own a breed that is fixed for this variant as well. I have Welsh Springer Spaniels. So it's part of what gives them their kind of long, low, rectangular silhouette. So what we do as a breed community, because this variant is fixed and there's not any breeding away from it, is we do pedigree analysis. So we look back and make sure that we don't have any relatives that have had back problems in the future or have had back problems very young in the future, to make informed breeding decisions based on that, rather than just looking at that variant.
Another question is, some of the issues in our breed are things that cannot be tested for yet, such as Addison's and epilepsy. Is there any new information that might allow testing for these disorders sometime soon?
I wish. So there's not any new publications that I'm aware of on either of those two diseases specifically. And again, it depends a little bit on the breed, but in that situation where we don't have a genetic test, there's no also phenotypic test. So when I say phenotypic test, I mean like OFA hips or elbows or, something along those lines. No phenotypic and no genetic test. We're again left with our pedigree analysis and kind of tracing back and making sure that we don't have relatives that are known to be affected, which of course is a little bit problematic because it does rely on other folks to be honest with you.
This person wants to know if there is a support person available by phone to discuss genetic testing results at Embark.
We do actually have a phone number that you can call. It is a voicemail line. I think we can provide you. I'm sorry, I didn't put it on the slide. I probably should have, but we can provide you with that. It is a voicemail line. So the way that it works is you leave a voicemail with times to call back, and then one of our results specialists can give you a call to discuss via phone.
Here's a question of how do we compare the COIs of a potential breeding?
So there are a couple of different ways to do that. We do have both matchmaker and pair predictor tools in our breeder platform. So one can only be used on dogs within your kennel. That's a pair predictor. And then the other, you can look at dogs from other kennels that their owners have said, yes, we can release results to look at through that matchmaker software. So we can estimate COI using that. Or if your breed is not eligible for matchmaker, there is a certain threshold that we need to reach of numbers of dogs tested and also released for results publicly. If your breed is not eligible for that, you are always welcome to write in to our customer experience reps, and they can actually calculate that by hand for you genomically. And there also are some pedigree softwares that will calculate it for you as well. Otherwise, you'll be putting pen to paper and doing a bunch of math.
Here's a question about ALT markers. She's seen many dogs come back with them on their test. Is there any reason why this is so popular? Also, can things skip generations?
So definitely things can skip generations, and that's typically what we see with recessive conditions. So often we'll have a bunch of dogs that are not affected, even though they'll breed for generations and not have any affected puppies, and then potentially will crop up later down the line. So recessive diseases in particular, whether we know what the genetics are underlying or not, do like to hide out. So that's definitely possible. And as far as the ALT marker, so that's actually another example of a personalized medicine marker. So just for some background for anybody who may not be familiar, alanine aminotransferase is a liver enzyme that's measured by veterinarians. It's just one of our markers for liver health. So about 50% of dogs that are tested with Embark do have that marker that is for low baseline ALT activity. So what that means is that those dogs are likely to have kind of a low normal ALT level just when we run routine blood work. So not a disease, not a reason to not breed, not a concern whatsoever, just important for your veterinarian to know. So the things that we like to do with those guys are, one, get a baseline blood level of that ALT when they're healthy so that increases can be interpreted more easily. And two, we're going to want to use other markers of liver health. So AST, GGT, ALP all come to mind when that dog is sick, because it is possible for them to have liver damage and that ALT still remain within the reference range. So it's not a problem for those dogs. There's no reason to toss them out of your breeding program. It's just good for veterinarians to know.
Great. Someone wants to know, can frozen semen be genetically tested?
Yes, it can be genetically tested. It is a bit complicated and also does require sacrificing a straw, but it is possible to do.
Someone else wants to know, when I test my upcoming litter, can I transfer the test results to the account of the new owner?
Yes, you certainly can do that. And I think we have a tutorial on how to do that on our website.
Okay, let's see. Next up, we have a question here about, oops, hang on, I just lost my questions. Do you have any resources to learn about coat color and how to breed for certain colors?
Absolutely. So we have some really, really great coat color and coat traits. So things like long hair, curly hair, furnishings on our blogs, on our website. So we certainly can link out to those. Those are great resources. And we also can do some pair predictions by hand if your breed isn't eligible for matchmaker. So if you write into our customer experience team, they can say, hey, about 25% of this litter is expected to have this trait, whatever percentage is expected to have this trait. So we can also do those by hand for you.
Another question is, if I test a puppy and the results is at risk, what that effect will have on the new owner's pet insurance?
That's a great question. So the caveat here is, of course, I don't work for an insurance company, and I can't promise that things will remain this way in perpetuity. But as of the current moment, since genetic tests are prognostic and not diagnostic, meaning they only show risk, they don't diagnose disease, they are not factored into insurance decisions. So they don't affect if a pet owner can get coverage, how much that coverage might cost. And in fact, it can also sometimes motivate your pet owners to sign up for insurance, just in case that does become a problem down the road.
Someone wants to know if there's any new developments on hemangiosarcoma markers.
Oh, I wish for that one as well. No developments in germline DNA or inherited DNA that I am aware of at this time.
Okay, here's one. Can genetic ancestry test to see in what generation admixture has come in when a dog whose breed is a single breed test with admixture? Can one see beyond a fourth generation?
So we can see back to essentially the beginning of time, if you will. We can reliably see back 3 or 4 generations, but as far as, like, when exactly that admixture occurred, I think that would be quite difficult to tell. Although reach out to me with your specific example, and I'll see if I can figure something out for you.
Yeah, please put that specific example in this question box, and we will get back to you with an answer for that. Here's another one. In dachshunds, are there modifiers to PRA, or do all affected truly go blind?
For the miniature long-haired dachshund, that example of the CRD4 cord 1 that I mentioned before, there are modifiers. We don't know what they are, but that mutation was actually initially discovered in a laboratory group of dachshunds and doesn't seem to have the same what's called penetrance. So as not as many in the pet population do go on to develop vision loss, or if they do, it does occur later than it did in that laboratory population. So there are modifiers, but we don't know what they are, and they're not testable at this time.
Okay, and then also do you happen to have the phone number to talk to a professional about test results on hand, or shall I get that for a later session?
We can reach out with it. I
Okay,
don't know off the top of my head, but I can find it.
okay, right. Okay, cool. All right, then. There's a bunch more questions, but they're kind of complex and deserve long answers. So I think we're going to wrap it up right now. Thank you, Jenna, very much for that presentation and for answering all those questions. We're going to meet back here at noon for our next presentation, which is I'm sorry, she's not Dr. Ame Llewellyn-Zadi, and she's from the International Partnership for Dogs. And her presentation, Science Made Simple, talking to new puppy owners about genetic health results. So we'll see you all back here at noon. Thank you.
Thank you.
Bye.